Dear students, welcome to EPG Pathshala. Today in this module, we will be discussing about the role of microbes and plants in remediation with special emphasis on rhizoremediation. Before looking into the learning objectives, let's introduce the topic. Now, you all know the soil is contaminated by a lot of many pollutants discharged from various sources, various industries and the domestic sources. Domestic sources from sewage and from industrial sources, it can be any type of industry starting from your chemical industry, pesticide industry, textile, distillery, tannery and so on. Now, the soil can be called as a sink of a majority of pollutants, both organic, inorganic as well as heavy metals. Now, these cause a lot of environmental and health hazards. Now, you have to remove or remediate the soil to avoid these consequences. Now, the remediation as such, if you see, you can do it by physical methods, chemical methods or the biological methods. But the physical and chemical methods, they are costly and they involve release of secondary pollutants. Now, also these methods cannot be applied in situ. They are ex situ techniques. I mean, the soil has to be excavated, brought and then treated and then returned back into the site. Now, bioremediation is a biological method. It is eco-friendly and it is cost effective. This can be applied in situ at the site where the pollutants are already existing. Now, as I told you, bioremediation is a process where the microorganisms and plants are involved. Now, within the plants, which is called as phytoremediation, in recent times a new field has originated called as Rhizoremediation. It involves plant-microbe interaction and it plays a role in remediating the pollutants. Now, with this introduction, let's look into the learning objectives of this module. Now, the learning objectives of this module would be 1. To understand the concept of remediation. 2. To identify suitable plant and microorganisms for remediation. To differentiate the merits and demerits of rhizoremediation, to study about the various factors that can be used to improve the rhizoremediation technique. As I told you earlier, natural and anthropogenic activities have led to the contamination of natural resources like your soil, air and water. It is essential to remove the pollutants from the environment. Soil being an important component of the ecosystem, they serve as a sink of all pollutants. The cleanup of soil is a challenging job and can be achieved through remediation. Now, physical and chemical remediation, they are not usually feasible because they are ex situ technologies, while your bioremediation or the biological remediation, they are a very good cleanup technique because they can be done in situ or on site. Now, bioremediation involves living organisms, mostly the microorganisms and plants, to degrade, reduce, detoxify or transform the pollutants into less toxic form. According to the biological agents used, the bioremediation technologies are categorized as bioremediation, phytoremediation and rhizoremediation. Bioremediation mainly focuses on your bacteria and fungi. Phytoremediation focuses on your plants and rhizoremediation, as I told you, they discuss about the plant-microbe interaction. Now, bioremediation uses microorganisms like bacteria, fungi, actinomycetes generally for degradation, removal or transformation of pollutant from the contaminated soils. Now, phytoremediation uses plants for removing the pollutants, and when the micro-plant assisted bioremediation approach is used, that is called as rhizoremediation. Now, this method or technology is gaining attention in the recent years. I mean, only it is just a decade old. Now, this Rhizosphere microorganisms, they play a role in remediating the pollutants. So, rhizoremediation is an approach where the microorganisms degrade or remove the pollutants in the rhizosphere by utilizing the amazing metabolic capabilities of rhizospheric microorganisms and the plant roots. Now, this term consists of both stimulation and rhizodegradation. Thereby, it is plant and microbial beneficial interaction. Now, the association between the plants and bacteria is an interesting area of research, and it has started since last decade. The term rhizosphere was first coined by Hiltner in 1904. Now, as I told you earlier, this is a zone which is present adjacent to the plant root system. Now, they provide a very active microenvironment for microorganisms growth, where through the root exudates the microorganisms flourish in the rhizosphere region. And these microorganisms which flourish near the root zone or the rhizosphere region, they possess very good capability of detoxifying or degrading the hazardous material. Now, this rhizoremediation involves certain steps. One is your identification and selection of microorganisms. Second, you have to select the polluted site. Third, to enrich the microorganisms in the pollutant concentrated area. Then it would be inoculation of the enriched microorganisms to the plant and root colonization. Then it is microbe plant host interaction, which would be used to clean that contaminated site. Now, let us see how you select a plant and microorganisms to perform rhizoremediation. Now, generally they say grasses and legumes can be the most suitable species for rhizoremediation, because they possess a highly branched root system which can support the growth of a large population of microorganisms. And these rhizosphere microorganisms are generally considered suitable because they possess a special property called as root growth stimulating or inhibiting properties. Now, let us look into the characteristics of plants which you will use for rhizoremediation. Now, the plants selected for rhizoremediation should possess certain characteristics. One, they should be fast growing and they should have high biomass. They should grow even under pollutant stress condition. They should not hinder the growth of plant. They should have a very highly branched profuse root system. They should facilitate the growth of more rhizospheric organisms, and they should possess an ability to accumulate metals in the above ground parts. Also, they should tolerate metal and accumulate them. The plant should withstand and resist the pollutant concentration. They should be easy to harvest and it should be non-consumable by humans and animals. Now, let us discuss about the characteristics of microorganisms which you will look into while using for rhizoremediation. Now, microorganisms should be capable of establishing a relationship with the plant selected for rhizoremediation. They should be able to proliferate in the root system of plants. They should have a catabolic pathway and they should possess certain plant growth promoting factors. Now, before getting into the further details, you should understand what is a rhizosphere and their structure. Now, Hiltner, who coined the term rhizosphere, he described rhizosphere as an area around a plant root that is inhabited by a unique population of microorganisms influenced by the chemicals released from the plant roots. Now, rhizosphere generally consists of three zones based on their relative proximity to their influence on the root. One is the cortex and endodermis, which consists of the endorhizosphere, where microbes and cations occupy the free space between the cells called the apoplastic space. The rhizoplane is the medial zone which directly is adjacent to the root, including the root epidermis and the mucilage. The outermost zone is the ectosphere, which extends from the rhizoplane out into the bulk soil. Now, the size and shape of the rhizosphere is not definable. It can vary or it can be as complex made by the microorganisms. The chemical, biological and physical properties of the rhizosphere, they keep changing along the root. Some places the microorganism population will be dense, some will be low. It keeps varying along the root region. The rhizosphere is always inhabited by a diverse range of microorganisms and is a zone of high metabolic activity. In addition to microorganisms, you also find many compounds present in the rhizosphere region. They are enzymes, sugars, amino acids, organic acids, vitamins, esters, etc. Now, from where does these compounds actually come? They are nothing but the root exudates which are generated from the plant. Now, these are used by the microbial population and then they grow and multiply. Now, Rhizosphere microbial population, as I told you, the size and type of the Rhizosphere or microbial colony generally depends upon the soil type, plant species, age of the plant, root exudates type and the concentration of the pollutant. The Rhizosphere contains both bacteria, fungi, actinomycetes, protozoans and algae. The rhizobiota use these root secretions and grow and proliferate in that particular area. Generally, these microorganisms or the Rhizosphere microorganisms protect the plant from pollutants and pathogens. Actually, if you see, there exists a relationship, like a symbiotic relationship between the rhizospheric microorganisms and the root. This figure shows the rhizosphere region of the roots. You can see on the left hand side, you can see a lot of bacteria, fungi and other microorganisms which are colonizing the root region, and you can see a lot of some secretions like mucilage which is shown there. The bacterial nodule, the bacteria once it enters into the root system, they sometimes certain bacteria they possess a potential to form a root nodule on the root. Now, figure B shows the internal structure of the root. You can see how the bacteria slowly penetrates into the internal root and then make an association with the root system. One is ectomycorrhiza and the other one is endomycorrhiza. Ecto is associated outside and endo it is inside. Now, let us discuss about the microorganisms which are involved in rhizoremediation. Bacteria. Now, the rhizobacteria that with certain plant growth promoting factors are called as plant growth promoting rhizobacteria. They protect the plant and they provide a defense system or defense property to the plants and protect them. Also, they possess capability of degrading the pollutants. Now, these bacteria they possess a symbiotic relationship and they are generally present intracellular between the root and the cortex cells of the root. Sometimes they are also free living and they exist extracellularly. Now, when it is associated with the internal tissues, they are called as internal or intracellular plant growth promoting rhizobacteria. When it is free living and it is present outside, it is called as extracellular plant growth promoting rhizobacteria. The symbiotic bacteria invade the plant cells. One example can be the root nodule bacteria which enters into the root cells and they form a nodule like structure which is a common feature of leguminous plants. Some of the examples of these bacteria will be Bradyrhizobium, Mesorhizobium and Rhizobium. These are free living rhizobacteria, I mean extracellular plant growth promoting rhizobacteria. Now, Agrobacterium, Arthrobacter, Acetobacter, Micrococcus, Pseudomonas, Bacillus and Serratia species, many rhizobacteria they do exhibit an unusual characteristics to survive under stress conditions. The stress condition can be a drought, salinity or heavy metal pollution. The rhizosphere are mainly dominated by the gram-negative bacteria. Pseudomonas is one example which has shown heavy metal resistance and they are isolated from diverse habitats. Next type of microorganism is a fungi. Along with bacteria, the rhizosphere is also inhabited by a lot of fungi. Now, they also possess a rhizoremediation potential. Now, the rhizosphere is mainly dominated by the genera Aspergillus, Fusarium, Penicillium and Verticillium. The zoospore forming fungi such as Actinomycetes, Pythium and Phytophthora are chemotactically attracted towards the roots by the chemical compounds secreted by the roots. Several fungi, namely Gibberella and Fujicorio, produce phytohormones which generally stimulates the plant growth. The next group of microorganisms are Actinomycetes, protozoans and algae. Now, in addition to bacteria and fungi, these 3 microorganisms are also found in the rhizosphere region with rhizoremediation potential. Actinomycetes are less stimulated in the rhizosphere region than bacteria. There are certain actinomycetes which possess antagonistic properties and sometimes they suppress the bacteria. These actinomycetes generally increase in number when the antibacterial agents are sprayed on the crop. Only during this time the actinomycetes increase in number when compared to bacteria. Among the actinomycetes, the phosphate solubilizing, namely the Nocardia and Streptomyces, have a dominant role to play in the rhizosphere region. Flagellates and amoeba also dominate in this region, unlike the ciliates. Now, let us discuss about the mechanism which is involved in the rhizoremediation. Now, there are 3 separate components in the rhizosphere. One is the rhizosphere region where the soil is there. It is a zone of soil which is generally influenced by the roots through the release of substrates that affect microbial activity. So, that is one zone. The second one is the rhizoplane. Now, rhizoplane, as I told you earlier, this is a root surface which includes strongly adhering soil particles. The third one is the root tissue, that certain endophytic microorganisms can colonize this area. The varying physical, chemical and biological properties in the rhizosphere soil compared with those in the surrounding soil are generally responsible for the change in the microbial diversity, metabolic activity in the rhizosphere microenvironment. Now, this phenomena is generally termed as the rhizosphere effect. The microbial population in the rhizosphere soil affect the heavy metal mobility and availability to the plant. They also degrade the strong hydrophobic organic pollutants by a mechanism of chelation, acidification and phosphate solubilization. Also, redox changes are brought about by these microorganisms. Now, let us discuss about what are the various ways by which you can improve the rhizoremediation efficiency. The biodegradative capabilities of these microorganisms and expression of these microbial genes in the rhizosphere are very important. Now, the other aspects that can improve the efficiency can be, you can select the best plant-microbe combination, you can select a suitable endophytic bacteria, seed colonization can be one way of improving, production of biosurfactants, and lastly engineering bacteria for rhizoremediation. Now, let us discuss each of this in detail. Now, selection of plant-microbe combination. The composition of root exudate in the rhizosphere changes with different plant developmental stage and plant species. Now, the variation in exudation can evidently impose certain effects on these rhizospheric community. Likewise, if you see, the root exudate composition also influence the proliferation of certain microbes which possess the degradation ability. Example, you can see flavonoids which are secreted by plants as a defense mechanism against pathogens can be one example. High amount of flavonoids can help in colonization of more microorganisms in the rhizosphere region. The environmental stress in the rhizosphere also varies the quality and quantity of the root exudates. Nutrient and water stress increases the root hair density. Likewise, phosphorus deficiency also increases the exudation secretion and it also affects the integrity of the membrane. These stresses resulted in increase in rhizobacteria population. Higher concentration of the contaminant can also affect the root growth and decrease the biodegradation of the contaminant. The use of bacteria with plant growth promoting factors can help in overcoming this problem. In recent times, if you see, the rhizobacteria with plant growth promoting factors attracted much attention in rhizoremediation. The root exudation quality and quantity will generally determine the fate of microbial population in the rhizosphere region. Rhizospheric microbial population can also help in promoting or inhibiting the plant growth by making the rhizosphere environment clean through degradation of the contaminant. Next way of improving your rhizoremediation efficiency is the endophytic bacteria. Now, Now, the rhizobacteria are more commonly used microorganisms in the rhizoremediation. But in recent times, if you see, there is certain endophytic bacteria which are explored for rhizoremediation purpose. They have concluded that the endophytic bacteria are efficient and advantageous due to the presence of certain characteristics. One, the endophytic bacteria inhibit the interior tissues of the plant and get protection from the direct contact with the contaminant present in the outside environment. They cause no negative effect on the host plant due to non-competitive nature of nutrients. It is easy to isolate an endophytic bacterium from the host plant, enrich in the requisite contaminant and re-inoculate the bacteria to the same plant for bioremediation. Now, these bacteria are also inoculated by various other methods like the seed inoculation, pruned root dip, soil drench and foliar spray. The choice of these endophytic bacteria inoculation method will depend on the specific type of plant endophyte combination. Endophytic bacteria from family Pseudomonaceae, Burkholderia, Enterobacteriaceae are commonly used in rhizoremediation. The endophytic bacteria are just in the infant stage, and the next method of improving the rhizoremediation technology is the seed colonization. The efficiency of rhizoremediation can be improved by using appropriate microbial inoculation method. It is one of the least expensive technique which cover the seeds with appropriate microorganisms. Here, for this purpose, the microorganisms should possess the character of adherence to the seed. The microbial cell adhesion to the seed has been studied by using viable cell counts and through reporter genes, especially GFP or Lux genes. The microscopic techniques like the scanning electron microscope has been used to track the microbial adherence to seeds, production of biosurfactants. Rhizoremediation efficiency can be increased by the bioavailability of the contaminant. Most of the organic pollutants are hydrophobic and they cannot dissolve in water. Now, these organic pollutants form insoluble complexes with soil particles and are biologically unavailable to the bioremediating organisms. Microorganisms use different approaches to promote the bioavailability of these hydrophobic pollutants. These approaches generally include secretion of extracellular polymeric substances, biosurfactant production and biofilm formation on the pollutant surface. Biosurfactants are amphiphilic in nature and form spherical or lamellar micelles when the surfactant concentration exceeds a critical micelle concentration that is specific for each compound. Now, hydrophobic contaminants become solubilized in the hydrophobic cores of micelles, which increases the transfer of compounds from solid to the water phase and thereby making it more accessible to bacteria. One important group of bacterial surfactants are the glycolipids, of which rhamnolipids are the major representatives. It has been shown that rhamnolipids are able to enhance the biodegradation rate of contaminants. According to Cooper and his colleagues, who isolated Pseudomonas putida strain from plant roots at a site from plant roots grown in polyaromatic hydrocarbon contaminated sites, produced two lipopeptide surfactants. Now, these lipopeptide surfactants increase the formation of emulsions with toluene. Now, this property is of great interest because a number of biodegradative microbes exhibit positive chemotaxis towards the pollutants. The combined action of biosurfactant and chemotaxis may contribute to bacterial proliferation and microbial spread in polluted soils, leading to clearing of ample zones. The last method for improving rhizoremediation is engineering bacteria for rhizoremediation. The application of genetically engineered microorganisms in increasing bioremediation efficiency has become a common approach. Now, these rhizoremediating microorganisms can be genetically modified by introducing a catabolic gene, construction of a hybrid pathway, promoter modification for increased expression of genes of interest and construction of recombinant strains. Now, the recombinant strains are the strains that possess 2 or more traits in combination, such as degradation of contaminant, production of biosurfactant, good root colonization capability and plant growth promoting factors. Still, the release of recombinant microorganisms is restricted in many countries due to certain legal limitations, which has limited its application or development in the field. Now, let us discuss about the merits of rhizoremediation. It is an in situ technique. It is more efficient than other biological remediation technologies. It requires less expenditure and energy. Also, it is natural and eco-friendly in nature. Rhizoremediation takes advantages of plant roots' natural symbiosis with mycorrhiza and root associated natural microbial flora for the enhanced degradation of pollutants in the rhizosphere region. It is easy to execute in field conditions. Also, they can be easily monitored. Now, what are the demerits? One, they are slow process. It requires days to months for removing the pollutant. For in situ remediation, soil must be of high permeability. It does not clean the site completely. Some concentration of the pollutant remains even after the process. Then, this mechanism of interaction between plant and microbe is still not completely explored. Now, what are the applications of rhizoremediation? It is useful for degradation of organic pollutants. It can help the accumulation of heavy metals in plants. It is also used for remediating the contaminated sites on a large scale. To summarize, at the end of this module, we have discussed about the concept of rhizoremediation. We have seen how rhizoremediation is different from other bioremediation. We have also seen what are the characteristics which are required for while choosing a plant and microbe. We discussed about the mechanism of pollutant removal using rhizoremediation. Then we discussed about various strategies used to improve the efficiency of rhizoremediation. Lastly, we discussed about the merits and demerits of rhizoremediation and application of rhizoremediation. I hope this module would be of help to you. Thank you.